Sehr geehrter Herr Vizepräsident, geschätzte Kollegen, Studenten, Angehörige. Als Leitungsmitglied des Departements für Informationstechnologie und Elektrotechnik der ETH habe ich heute das Vergnügen, die Einführungsvorlesung von Professor Janos Vörös anzukündigen. Professor Vörös' Forschungsgebiet ist die Bioelektronik und Biosensorik und markiert eine weitere Expansion der Life- und Bioscience an unserem Departement, das schon eine lange und erfolgreiche Tradition in biomedizinischer Technik hat. An der ETH ist die Verbindung von Ingenieurwissenschaften und Biologie und Medizin schon seit etlichen Jahrzehnten vor dem kürzlichen Bio-Nano-Hype geprägt worden. Die verstärkte Verbindung zwischen biologischen und elektronischen Prozessen auf der Mikro- und Nanodimensionsskala bringt uns vielleicht zu einer Wende, ähnlich wie bei der Halbleitertechnologie vor etlichen Jahrzehnten. Lassen Sie mich auf ein paar Meilensteine auch in der Karriere von Professor Vörös hinweisen. Wie gesagt, der letzte ist, wenn ich dem so sagen darf, er ist seit dem 1. Januar 2006 außerordentlicher Professor für Bioelektronik an der ETH und gehört zum Institut für biomedizinische Technik. Professor Vörös wurde 1970, wie Sie am Namen sehen, in Ungarn geboren. 25 Jahre später, wenn ich den großen Sprung mache, 1995, hat er den Master of Science in Physics an der ETH-Zurich in Budapest erworben. An derselben Universität doktorierte Janos Vörös Biophysik im Jahr 2000 mit der Promotion zum Thema Optical Waveguide, Lightmode, Spectroscopy in Biomaterial Research. Schon während seiner Ausbildungszeit in Ungarn hat Professor Vörös Verbindungen zur ETH-Zurich geknüpft. Von 1995 bis 1998 war er Forschungsassistent bei der Firma Vacuum in Ungarn, ich denke während der Doktoratszeit sogar, und hat auch während dieser Zeit, 1998 bis 2000, hat er die Position des wissenschaftlichen Mitarbeiters am Departement für Materialwissenschaften der ETH inne. Nach dem Doktorat folgten dann ein paar Postdoc-Jahre an der ETH zwischen 2000 und 2003, die anschließend in einer Stelle als Forschungsleiter mündeten. Zu Beginn des Jahres 2004 wurde Professor Vörös Leiter der Gruppe Dynamic Biointerfaces am Laboratorium für Surface Science and Technology vom Departement für Materialwissenschaften auf der ETH Zürich. Zusätzlich zu seinen ETH-Verpflichtungen amtete er seit November 2005 als Adjunct-Professor an der Pennsylvania State University in den USA. Und wie gesagt, seit 1. Januar mit einem Jahr noch, mehr als einem Jahr ist er außerordentlicher Professor an der ETH und leitet die Fachgruppe Biosensoren und Bioelektronik. Sie werden jetzt sehr viel Bio hören, wenn ich seine Interesse aufzähle. Seine Interesse liegen, wie gesagt, in deren Forschung im Bereich der Bioelektronik, Nanobiotechnologie, Biosensorik, Biophysik und Biomaterialien. Es ist fast überflüssig zu erwähnen, dass Professor Vörös über eine imposante Liste an Publikationen verfügt und auch Mitglied etlicher schweizerischer und internationaler Fachorganisationen ist. Er ist Inhaber mehrerer internationaler Preise, die ich hier nicht alle auflisten möchte. Professor Vörös wird uns nun mit seinem Vortrag Bioelectronics, Interfacing Electronics with Biology at a Nanometer Scale Aspekte und ich denke auch Herausforderungen seines Forschungsgebietes nahebringen.
Thank you very much for the introduction, and it is a real pleasure to see so many familiar faces on this beautiful spring afternoon. And I really hope that you will enjoy my tour through bioelectronics, which I would like to convince you that we arrived to a new era where we can really interface electronic devices with biological entities, because we can control such biological objects down to the nanometer scale. And these first two images, you see two complicated objects. One of them is a biological cell, and the other one is a microprocessor. And of course, they are extremely complicated, both of them, but they are very different. A microprocessor is, we call it artificial because it's engineered. Each of its elements is performing a function which we define, and each of its parts is in a place where we want it to be. A cell is natural, and partially natural means that it's self-organized. It builds up itself, it controls itself, and it consists of small nano-sized objects, similarly to this one, but these nano-sized objects self-assemble and interact with each other in a very different way than this defined feature here. And I would like to convince you in the next 45 minutes how we can combine these two and how we can use processes which we learn from the biological object to develop artificial engineered objects like this one here. Or how we can use processes that we develop to create that microprocessor to address the cells, the biological objects, in a novel manner. You see here an example. This is the way we produce a top-down structure. It's not because my son is playing for the ZSC Lions, but this is just an illustration that we can produce a top-down structure where the biological entities self-assemble in a predefined manner. Of course, this is on the macroscopic scale, but I hope that I will be able to show you how we can do it down to the nanometer scale. So let's quickly look at these length scales which I was talking so much about. If you look at bioelectronics and biosensors, the normal devices which you see at the everyday life, they are in the human length scales. If you look into the elements of biology which connect to these devices, neurons, these are in the 100 micron length scale, or the spots of a microarray are also typical in this 100 micron. And if you go below, you look at an individual synapse, a connection between two neurons, or you look at the carriers of life, vesicles, they are already 100 nanometer. And finally, you reach the size of proteins. And these proteins are the building blocks of life. And this is which we would like to connect to artificial devices, and we have to do it in an ordered manner that they preserve their conformation, orientation, and they interact with the artificial device in a well-defined manner. And we live in a good time, because we do have a variety of technologies which allow us to do that. You can see here a list of top-down approaches and some bottom-up processes. And if you look at this table, you notice that between the micrometer and the nanometer scale, you have a variety of tools that you can combine and use to address these length scales. So the message I would like to deliver today, the main message, is that if you can control the bioelectronic interface down to the scale of the individual building blocks of life, then you have the chance to control the function of the device, be it a sensor or a biomaterial or an implanted device. This slide summarizes what we try to do in the laboratory of biosensors and bioelectronics. Here, I listed the expertise we need to develop enabling technologies I call bioelectronics, micro and nanostructuring, electrochemistry at the surface, physics and chemistry down to the nanoscale. We don't do any polymer synthesis ourselves, but we collaborate with good people who can do all kinds of useful polymers, which we can then implement to our systems. And where we would like to be strong is applying all these tools to sensing approaches, to interfacing biology. What I mean by interfacing biology is controlling the growth of neural networks and controlling the interaction of cells and materials. And then also in the biomedical field, well, we develop tools for local drug delivery and recently also some new approaches for tissue engineering. So what do you need to do that? First of all, you need a good team. And I'm really happy to show all these people who are part of my team in the laboratory of Biosensors and Bioelectronics. And it's really this fantastic team which allows me to be here today and talk about things which we have done during the past year. They built a lab, they are running it, and they do fantastic stuff there. So let me show you a few examples. But before I do that, it's not enough to have your own team, you also have to operate in a network. And it's really nice to have so many different colleagues within the ETH. It's good to be having such wonderful hosts at the Institute for Biomedical Engineering and in the ETH. It's also good to have so many colleagues from ETH itself or within Switzerland. And recently we have a growing network, an international network through various European projects or international collaborations. And last but not least, it's also really good to have a support of the family. Without that, it would be very difficult to do the work of a scientist. And you can see that my children and my wife, Andrea, Anna, Zsófia, Janos, and Katalin, they helped me to discover what is behind the mist of science. So what kind of tools we have in the lab to control the biological interface? And first of all, I'm going to show you some tools that allow us to control this interface on the nanometer scale. And then I will move to this beautiful interplay between electronics and biology. How we can use electronic control and light-based control to perform tasks which were not possible before. Let me briefly talk about some polymers. This is a famous molecule which we discovered when I arrived here in Switzerland, originally synthesized by Jeff Hubble and Don Albert. It has a PLL backbone, which is positively charged at physiological pHs, and polyethylene oxide side chains, which then bring protein repellent properties to the molecule. And this molecule makes surfaces resistant to all biological interactions. What you see here is a titanium oxide surface in a biological solution serum, and you rapidly see a development of a protein film on it. If you coat this same surface with a layer of this PLL PEG, then no proteins will bind. And it's very strange because I'm always talking about that we want to control the interplay between biology and devices, and I yet talk about devices or surfaces that don't interact with the biology. And the reason is such that in order to eliminate all these non-specific effects and you define specific functions to surfaces, you really need to have first the repellent substrate. And then you can think about introducing functions. So here you see the different functions which were developed by the Laboratory of Surface Science and Technology and now commercialized by Surface Solutions. So we can buy all these molecules with various functionalities and at the same time having properties which repel all the non-specific interactions. So let's see how this could work. You have a biotin functionality on this molecule. You stick your surface to the solution. You will get rapidly the molecule assembling there. You put in serum full of different proteins. There is no non-specific interaction. And when you put the binding partner of this biotin group, streptavidin, then you will see a rapid response and you will get what you wanted to have from the device. And you can make it more complicated. You start out with the resistance substrate. You use this neutravidin or streptavidin as a glue to stick a single-stranded DNA to your surface. And then you put the complementary strand to a nano-sized object, let's say a lipid vesicle, which can carry all kinds of functions. And then you let this self-assemble and bind to your substrate. Here you see the process, how it happens online. First, the PLL PEG binds, and then the complexes of neutravidin and biotin DNA, and then the DNA-tagged object, nano-size object. And why would you do that? Because in lipids, in such vesicles, you have membrane proteins, and these membrane proteins are the most important drug targets. And we know very little about them. Because they are notoriously difficult to purify, they are very difficult to put onto our devices. And the reason why they are difficult to put onto the devices is because they only survive in their natural lipid environment. So we need to have some tricks to bring them down to our sensors, for example. And one of the tricks would be to use a DNA array and convert it to a membrane protein array. How this would work? Simply, you can buy these DNA arrays now. You can produce them, the whole human genome on a chip if you want to. And then what you do is you define the location of certain membrane elements with the DNA on the chip, and you tag a lipid vesicle which carries that membrane protein with the complementary strand, and they would self-assemble according to the pre-given structure. And in this example, we have done it with microfluidics, but we can also do it just self-sorting on a spotted array. What you see here is that we define with one DNA the position where vesicles which carry a green membrane protein should bind. And we define with another DNA where the vesicles which are tagged with a complementary DNA carrying a red protein should bind. We mix the two, and what you see here is that they sort on the letters of ETH according to the pre-given location of the DNA there. And this is a very interesting concept. We define the position of nanoscale objects with the DNA. And the DNA will take care of all the non-specific issues, will take care of the specificity of the system. Let me show you an example how we do patterning using self-assembly. What you see here is a very simple approach. You pull out a sample from solution which contains micro or nanoscale particles. And when you do this pulling out, you assemble the particles on the surface, you do a reactive ion etching step, you shrink the particles smaller, and then you will have nano-sized features sitting on your substrate. And you can control very nicely the distance with the particle size and the size of the features with the etching time. So here you see a little bit the homogeneity which you can reach. You can make beautiful hexagonal patterns of particles. You see this is 1 micron, so we are far below the micrometer scale. Then you can make much larger arrays. You start to see the domains here, but it's still working. And this is very useful because it's a simple process, and you can then produce nanoscale patterns with cheap and dirty technology for biology applications. And you can also functionalize them. With the little tricks, you can achieve that proteins will only sit on the dots, and you can code the background with the protein-resistant pattern and think about using these for addressing issues, how cells interact with such nano-sized features. I also told you that we can combine the top-down approaches with bottom-up assembly. And let me show you how that works. You start out with a simple lithography step. You produce on a whole wafer a photoresist. Then you functionalize the open areas with a molecule which carries the functional group. You lift off the resist, and then you backfill with the resistant PLL peg. To show you that it really works, here you see 5 micron spots of streptavidin sitting in a protein-resistant background, and you can really control the ligand density on the spots while keeping the background resistant. Or you can convince cells that they should interact with the surface according to the pre-given pattern. Here these cells follow the squares, not because that's their natural way of formation, but because we provided on the cells ligands, RGD peptides, that interact with the cells specifically, and they don't find anything else on the substrate. So they really grow where we want them to grow. If you have good collaborators, for example, Harun Solak in the PSI, then you have access to nano patterns. This is an X-ray interference lithography technique. Here you see one of such nano patterns where the dots are about 100 nanometer in diameter, placed 500 nanometer apart. This is the photoresist pattern, 30 nanometer. You functionalize the dots with the molecules of interest. You remove the background, and then you reach 2.5 nanometer dots, thick dots sitting far apart. You backfill. You add your glue streptavidin with the DNA. And in this case, what we use is gold nanoparticles, tagged with the complementary DNA. And again, the DNA defines the position of where these nano-sized particles should assemble. You see that they are nicely assembled only on the dots. And it's a dip and rinse process, essentially, after the lithography step. So you can really do it in the lab. This is now 2 micron. You see these nice nanoparticles sitting on your pattern. And you can think about putting 1 down, or 2, or 3, or 4, or if you make the pattern a bit larger and the particle smaller, then you can make 32 on average on 1 dot. And why would somebody be interested in doing this? Because this way, we can use the special optical properties of these nanoscale objects for sensing purposes. Because the absorption of these nanoscale objects, the visible absorption of these nanoscale objects depends on refractive index conditions around these nanoparticles. And this refractive index changes when something binds to these objects, and that's a sensor. So we really try to develop this towards a simple sensing, optical sensing tool. But if you can produce dots of nanoparticles, you can also produce lines of nanoparticles. So what you see here is micron range wires produced with, from nanoparticles. And of course, if you use these nano-patterning tools, then you see that the particles assemble 1 by 1 on the nano-size line. And if you add a little bit more of the particles, and you do a little bit of gold enhancement chemistry, then you can achieve conductive nanowires. What you see here is about 100 nanometer self-assembling nanowires. And this is one of the elements of a microprocessor. We can produce conductive wires by defining the position of the particles with the DNA. And if you don't use one type of DNA, but you use different type of DNA, you will have the chance to produce self-assembling processors. We will not only use gold nanoparticles, but also some semiconducting particles, and we will try to assemble transistors from solution. Very different concept than the normal electronics. Of course, we try to use it first for sensing purposes, because the conductivity of these wires depends on what binds to their surface. It's so thin that it acts as a transistor. The gate itself is a biomolecule which brings a charge, and the resistance of the wire will then change due to the small size upon absorption of a biological object. And it, of course, generates a lot of attention nowadays. This is the front page of the MRS bulletin recently. And we try to achieve such new sensing devices. And why would we want to do that? Because if you look at what happens in the hospital with a blood sample, what you see is it goes into a centralized lab where you can have about 800 samples per hour. You can measure over 100 different parameters with high precision. But you need to carry the blood sample to a centralized lab, and you need quite a bit of blood to use. On the other hand, we already have devices which measure the amount of glucose from a drop of blood here. So here you see a drop of blood, and this device would measure the amount of glucose. Can we merge the two technologies? Can we reach that this whole large robot would be squeezed into such a small device? The only limiting factor, or one of the most limiting factors, is that we have to be extremely sensitive. And with the help of these nanowires, or with these nanoparticle-based optics, we might be able to reach the sensitivity which required to multiplex all this robot into such a small handheld electronic readout device. Let me show you just one story about a typical lab day. It's not a typical lab day, but it's a very interesting one. So we were sitting with a student in front of the microscope, and what we wanted to do is just control our nice patterning tools that we can bind nanoscale objects to certain locations. And it is absolutely normal to as a control experiment, to photobleach certain areas of your fluorescently labeled molecules to see that the intensity is there and there is not much non-specific adsorption in the background. And what we noticed, that where we photobleached, like this smiley, there the nanoparticles didn't bind. And this was something very strange, because of course, when you do photobleaching, you in principle don't do any destruction except that you kill the dye. But here what happened, we not only killed the dye, but locally we also suppressed the hybridization of the DNA. We generated reactive species, which later on turned out damaged the DNA. And you can use this damage actually as a tool in biotechnology, because now we can use a protein, a fluorescently labeled protein, as a photoresist. Here we illuminated light locally. The protein is damaged, goes away, and then we come with another color protein and put in something else there. And since we use these molecules to define the position of different objects, this is nothing else than a biological photoresist. Let me also talk a little bit about electronics. The molecule I mentioned earlier has a positive charge on its backbone. So it gave the idea right away what happens if you apply potentials. You need a device to sense it. We use an optical waveguide-based device with an electrochemical cell on top. And then you see what happens with the molecule when you apply potentials. At 0 volt, the molecule assembles. When you rinse, it's stably there. You apply a potential, you can completely remove it. And then you can put it back again. And this is very nice because it opens up a variety of possibilities for us. First of all, you can readily buy indium tin oxide microelectrode arrays. These are transparent. Each of the dots are individually addressed electronically. You can put this array into a confocal microscope, connect it to a switchboard, and then to a potential start to control the individual dots electronically. And the concept is very simple. What we do, we take the bare indium tin oxide and the neighboring resistant areas. We coat it with a fluorescently labeled resistant molecule. And then we activate certain spots. What you see here is a movie when we do that. So where the arrow shows up, we apply the potential, and you see the red color fading away. Because the red labeled molecules go away, we zoomed into the 5th spot, and then you see when we apply the potential, we remove the molecule. It's very fast, and we end up with a bare substrate locally there. And this is really nice, because now what we have is we activated certain spot, and we can put down, of course, something else there. And why not putting down a lipid bilayer again? Because of the relevance to membrane protein screening. So what you see here is these 2 dots were already functionalized with a green labeled, fluorescent labeled lipid. And when I start the movie, you see that first we activate the 3rd spot there, and then we add the lipids in solution. So everything is still present in solution. So the green labeled lipid vesicles are coming, and then they form a bilayer also on the 3rd spot there. So now what we can do, we can functionalize the different spots of an array with different molecules. And these can be channels, which is very nice because it's directly on an electrode, so you can monitor channel activity. These can be receptors, let's say taste receptors, and make an artificial taste sensor for Coca-Cola, because we have only 450 of them, so we can put all of them onto one chip now, because we have the tool to dispense them locally. And we can also control the growth of neurons locally. And this was always a concept which generated a lot of attention. Nowadays, what we do, we interface with electronics through our hands. But of course, there are many applications where it would be much nicer to interface directly with the brain. One of the serious ones is to help patients with disabilities and help them to control motion and machines with their brain. I believe it's really challenging, but a lot of people try it, and Peter Fromherz was one of them. So you see, for example, here neurons which grow onto the chip, but what you see here is that they grow where they want and not where we want them to grow. This is also one approach where you put brain slices over the electrode, and you try to understand how these sliced brain interconnected and what is the communication between these neurons there. A little bit similar to cut your car into 100 micrometer slices and trying to understand what the connections are there, but I think it's 2 orders of magnitude more difficult. So I put a much simpler challenge in front of me. What we would like to do is we would like to control the growth of neurons on the chip from the beginning. And we want first 1 neuron, 2 neurons. And here you see that we have already reached a certain aspect. The neurons are growing only in this area, and on this electrode they don't grow. On this electrode they don't grow because we control the surface chemistry locally with the potential. So this electrode is buried under the cells because we remove the protective coating there. While these electrodes, there is no cell growth. Here you see it with many, many cells. What you see here is how you can control it with a single cell. This neuron is sitting now on a different channel, micro-manufactured. Next to this, there are electrodes which prohibit the growth of this neuron coming to this direction. On the second one, we let it grow, and then the neurites are growing. On the third one, you see the first attempt to connect 2 neurons together. So basically, we have now the tools to control the growth of neurites and neurons on the chip into different directions. So we will be able to build small networks of neurons and then start to study them and from the bottom up. Let me talk you a little bit about electronically controlled drug delivery. This is a major issue because for many drug delivery devices, it would be useful if something you can be measured, and then right away you could administer the drug and dose it according to the need of the patient automatically. So what you see here is an old slide from science where you build up layer by layer positively and negatively charged polymers. So we did the same. We chose PLL, polyl-lysine, positively charged, and heparin, a model drug, and we built it up layer by layer on our substrates. So you can build up a multilayer. And the beauty of this multilayer approach is that you can put in all kinds of different biologically relevant molecules. You can have DNA, proteins, vesicles, neurotransmitters. And when we apply the potential, you see that it's nicely released. And you have a complete electronic control. When you stop the potential, nothing happens. When you apply it, you release. Stop, nothing happens. And this is, again, a new way of interfacing with biology, because now the electronics doses what we release and where. Just let me speculate a little bit further. We can control, of course, down to the micrometer scale of where our electrodes are. We can build and embed all kinds of active elements to these multilayers, and we can control the growth of cells over the surface where we want them. It's nothing else than artificial synapse. This is exactly how the neurons are communicating with each other. One thing is still lacking. This will be depleted after time. This is constantly remodeled. The last example I want to show you is electronically controlled tissue engineering. Here you see a movie where researchers in Japan harvest a layer of corneal epithelial cells from the dish. What you do, you take out from the patient's eye, you take out a healthy cell, you grow it on a Petri dish, and then you can see the transparent sheet there. You see it also partially here, and implant it back of the eye of the patient. This is a rabbit in this case, but the rabbit got back its sight completely after this surgery, because these are autologous cells. In the next example, it would be really nice to do the same, let's say, with heart attack damage. If you have a damaged part of the heart, you can harvest healthy heart muscle cells, grow them in a Petri dish. You see the beating cell sheet here, and then put it back on the damaged area. And you can get back the performance of the heart. And it works beautifully. You get back 90% of the performance lost of a mouse heart. But unfortunately, it only works for the mouse and not for the humans, because so far we can only stack 4 layers of cells. If you put too many on together, the layers, the intermediate layers don't get enough nutrition and oxygen. So it requires more engineering approaches to achieve that we could also keep thicker tissue constructs alive. So we thought to take our approach and use a sacrificial layer between an electronically conductive substrate and the cells. And what you see here is that the cells grow on this polyelectrolyte multilayer, and when we apply a potential, then the cells are harvested from the substrate, and you end up with a cell sheet. And again, let me speculate a little bit. Because we can control locally what happens and where the cells can grow and where not, we might be able to control, like initiators of blood vessel formation, might be able to deliberately engineer porosities into these sheets, control two-dimensional arrangement of cells, and then harvest the cell sheet and put it into a tissue construct. So as a summary, I showed you how we can use self-assembly approaches to address electronics, how we can use microelectronics to build new type of sensing devices, how we can control the growth of neural networks, and a little bit also what we could do towards tissue engineering. I came to the acknowledgements. I would like to start with my parents. Without them, of course, I would not be here. And I always got their full support. They are also here somewhere in the audience. And then I would like to also acknowledge all the teachers who helped me to get here, because you tend to forget them, that the career of a scientist starts in the elementary school and then later on in the secondary school. And this is the old building where I started my studies of my university. And in the last 2 years, during my PhD, we already moved to this new building on the side of the Danube. I would like to also acknowledge the biointerface group of the Laboratory for Surface Science and Technology, and especially Marcus Textor, because he was a real mentor for me, and he really helped me to get here where I am now. And once again, the fantastic team which does all the work in the lab and helped me to stand out here and talk about these scientific discoveries. It's not only the students, but also the technical staff and the secretary, Esther Zinger. I think we all together make this fantastic team, and it's really fun to work with you all. I would like to also acknowledge the different funding sources which help us to perform all these expensive experiments. And finally, I would like to thank you for your attention and invite you for a little apéro. And these two wines, one of them is Bull's Blood from Eger. This is a famous Hungarian red wine. And the other one is Cserszegi Fűszeres, which is just known around Western Europe as the wine with the unpronounceable name. And actually, it was a professor in my hometown who bred the grape which this wine comes from. So I really hope that you will enjoy it. Thank you very much for your attention.